So I'll go ahead and I'm actually going to record this one because I got some new stuff in here. It'll be fun. So, who knows that title reference, by the way? Raise your hand if you're cultured. Ah, yeah, there you go. Nice. That's fine if you didn't. It's fine. Don't worry. It's worth reading sometimes. But anyway, the AI overlords have given me DNA, cells, and drugs in this case, and that's kind of the outline. But like Greg said, I do want to kind of explain myself. Yeah, here's where I'm from in Colorado. Pretty fun. Hanging Lake. Google's it. You need a permit now. Sorry. Don't jump in that, by the way. Went to Carleton. Super fun. A lot of nerdy people, but they were nice. It was cool. Omaha, super underrated. So if you guys are thinking of more grad school, UNMC's got a lot of money. Warren Buffett's still around. It's a good time. And then, yeah, Mayo Clinic, Rochester, and then here. Because even though the science that I was able to perform here and here was super impactful, really liked the field, I did want to go back to teaching and kind of be part of that life again. So let's go back. Let's take a little lesson of some immune cell type stuff. So everybody in the room's been sick, right? Everybody's had a bit of a go. Who had, let's do this quick here. Who's had COVID? Yeah, I think pretty much everybody. I'm gonna make a guess. And eventually, all of us are here. It cleared out, right? And now you have memory against it. So the whole system that we're kind of gonna be setting up here, based on something called a B cell. You have immune cells that exist inside your white blood cells, or sorry, well, they are your white blood cells. They have this little receptor right here. And when the time is right, pieces of virus or bacteria may encounter this receptor and may be able to bind to it and say, this looks like something that I recognize with that little Y-shaped receptor right there. Should they be given the right signals, you end up with a B cell that we kind of typify with the antibody response. Those little Y-shaped guys are a set of gene segments that form this nice setup right here. It's going to bind antigen, pieces of pathogen that you want to get rid of. Now, this is all good. This is how our immune memory becomes and kind of has that massive strength. The cool thing with the plasma cells, when you guys have been sick before, remember when you got better, it was just sort of a overnight period, right? You woke up and it was gone, right? It's because once that plasma cell shows up to the game and it binds the target, it floods your bloodstream with quadrillions of these things. But to say the least, the path to get there is a little tough. What if I told you that? And well, to remind you, none of us had ever had COVID before. It was completely new to your immune system. It had never been seen. Your body evolved a response in real time, the same way that if you went to the Amazon jungle, jumped in a pool of water, and got a parasite that no human had ever seen, same thing would happen. You have these little mature naive B cells. They have what I'm kind of highlighting, the antibody actually starts as a receptor. They essentially exist in your lymph nodes and are kind of piling up a little bit until they get a signal. If they get the signal that something is incoming, some bad thing, they actually go through something called somatic hypermutation. See up here, this is a little gene right here. Oh, I can't even zoom in too much. But to make that gene a little more unique, this is the one area of your body that actually mutates itself. Now, the reason is, those of you that are more ecology and evolution based, it's because it is forcing the hand of a new strong, basically organism in this case, but out of the population. So should all of these characters be around a T helper cell, a dendritic cell, what those jobs are is essentially to say, not only bring pathogen, but say, this is the right thing to kill. I'm going to train you to kill this thing. Should the B cell right here bind to the same target and pathogen identified by these initial helpers, you're in business. And the B cell is going to be told, hey, we want you, the one that could bind to our target. We want you to grow. We want you to survive. Seems good, right? It's the one that can kill this thing. It's going to go all the way back. It's going to redo the cycle. This cycle will repeat 3 to 4 times. This is why when you're really sick, it takes about like 3 to 7 days to get better, right? This cycle is happening. You are evolving a response. Now, the kind of the animal ecology thing that I always think of here is that eventually, at the 3rd or 4th cycle, only one clone eventually beats the others because there is a competition for resources. And the resources, the food in this case, is the amount of attraction and signal. This little antibody binds to that horrific pathogen that you want to kill. Eventually, one wins. The animal example I always think of is, you guys have heard of that shark species that only births one because that one was the one that ate the others in the womb. Outcompeted all of them. Yeah, lymph node time. Regardless, eventually you've got a winner. Plasmablast shows up. This gets our plasma cells. We are going to be booming these out. Business is booming, as crazy Antonio Brown might say. Now, you'll also have some cells that will go into stasis. They will wait, and they will be there ready right away. Now, the good news is we don't have to suffer the slings and arrows of smallpoxes, COVIDs, and other things. We have vaccines that can train this response to be a little more light to the touch, to say the least. But in all of that, you catch a few things about this process that might go bad. The main signal that these cells are constantly competing for, grow and survive. Cyclic mutation. I know, I even forgot my favorite, this class switch recombination. You're going to be switching and splicing and moving different genes to the right spot. So cutting things, mutating things, growing and surviving. It's kind of starting to sound a little like tumors, right? Surprisingly, lymphomas, cancers of the white blood cells, are about the 7th most diagnosed on the planet, actually. But it kind of makes sense, right? These things are going through a constant state of flux and anger. So what we do and sort of what carry on in the field, and I'll say we, even though we're kind of a small fish in this case, first aim is we have tons of publicly available patient data. This can range from DNA alterations, RNA transcription up and down, and eventually we'll see some single cell RNA. So not just a bulk sample of RNA, but every single cell has its own profile, things like that. And we can say, using identify, which patients are not doing well, which genes say that you're going to fail the therapy. And we can take a look at those patients. We can find them, isolate them, and maybe even find the exact signals that are driving those issues. Now, second step, should we find in the bioinformatics, quote unquote, our little, some stat finding, let's say we want to go investigate. We have cell lines, living human cells that exist in culture and incubators. We can extract their genes, the proteins in this case, and investigate. In this patient, DHL6, or this patient, U2932, does this gene exist in higher or lower levels, and at what amount? So in this functional validate aim, number 2, we would like to see what is driving this pathway. Is it actually there in the real life cells? And in my case, I don't enjoy working with mice too much. They're inconsistent, and I don't want to kill all of them because I'm making mistakes or something. So luckily, human cell lines can be a very good model for that. But the third and favorite, and if you asked any of my research minions, they will tell you the favorite thing is to take the cells and put drugs against them and see, given what we identified over here, is there a good drug against that gene that will kill this stuff? And the cool news is, we have so many drugs out there right now that there's not even enough people to test all of them. That's where we can kind of come in and say, and the reason we use the word translate here, it's going to be a bit of a theme. And it's how I was trained at UNMC. I was trained by a PhD set, but an oncologist and a chemist as well. Translate is that you have to bridge that gap between the essential science, but you have to give something that the clinic can use. Basically, the philosophy of my PhD mentor was you can't just stop the science at that point. You have to try and bridge that final piece, because otherwise it can take 10 years to get that far. All right, so who all here has had a family member of any kind experience cancer, right? And basically, again, every hand theoretically is going to go up. 1 in 3 about will encounter some sort of cancer in our lives. These are what we call hallmarks. They're pieces that we typically see, features that sort of convergently evolve, if you want to put it that way, in most tumors. Now, if I had to be pretty strict about things, you probably need this one. You need mitosis. That's got to happen. You need more. Basically, 1 cell has to become 2 over and over again. That's why we hammered mitosis into you all in 1 of 5. Now, among these others, though, it's where genes kind of start showing up. If you can delete growth-suppressing genes, lose them, mutate them, it's only going to help your case. Once the guards are out of the prison, the prisoners run the game. There's one on here that is probably more important than the others in terms of a true bad one. And a tumor is bad if it multiplies a lot, but a tumor that doesn't die when you drug it or try to kill it is a rough setup, okay? Here's one way that they don't die. Constantly, your immune cells right now are hunting and finding potential cancer cells. Probably all of us in this room have had 1,000 precancerous cells killed by your immune system. And they caught it and nipped it in the bud. It's the power of your immune system. But when tumors do show up, they formed some partnership here. And this avoid immune destruction is going to be one of the more fun aspects of what we're kind of up to in our research this round, which is exciting for me. So, get into some fun basics. You guys remember Bio 211, right? Here, we're going to put a little, there we go. Yeah, we love genetics in here. All right. Should you have a normal gene, everything's fine, hooray. Most of the time, when you mutate, even a point mutation like that, you're going to screw stuff up, or you're going to mess stuff up so badly the thing just can't even exist. It gets trashed immediately. And the opposite of this coin would be gene mutations that accidentally keep a gene on, a growth gene, and it never stops potentially. So there's a two-way street on this. But in most cases, we can identify these. We can sort of predict these a little bit. They offer some, I would say, they offer us some sort of like kind of cash on the future of what's going to happen. Eventually, with the right setups, you eventually stop going this way. Apoptosis is a process where a damaged or messed up cell is sacrificed essentially on its own, and it flows up. But it's ordered. It's part of the natural system. You would much rather lose one bad cell than let it go free. Sort of guilty till proven innocent in that case. Because if you let one get out with damage, cells become less like cells of a multicellular organism. They start to behave more like a parasite, an animal that runs around and moves and does its own thing to survive. It's very hard to kill. Those of you, I think, even working in Dr. Worsel's lab, how hard is it to kill an invasive species? Not ideal, right? Something that grows faster and dumber than the other native species. So that brings us to the disease finally. It's a big mouthful, but it's the most common white blood cell cancer. It's the most common lymphoma. Diffuse large B-cell lymphoma. If we went back to this section right here, diffuse large B-cell lymphoma is basically just the meat of this cycle, that middle cycle in there. There are other lymphomas that can attack the other ones or come from the other ones. But given that we are mutating and splicing our genes in this case, it makes sense. But those of you have probably heard of stages of cancer, right? Stage 1, 1 node, stage 2, 2 nodes, but in the same tissue. Stage 3, same organ system, but multiple nodes. Stage 4, that's the one that we typically can't treat. Spread into the lungs, into the brain, somewhere else. You can't win a war on multiple fronts. We have to prevent things, typically, from ever getting there. But here's the problem. Most cases in most cancers, you can surgery out, right? In a lot of cases, right? If you have a colon cancer and you catch it stage 1, you can get that out of there and it's cured. Can you surgery out your white blood cells? Now, that's the problem. So in the leukemia lymphoma fields, there has always been a necessity of experimentation. There has always been a necessity of chemical agents to try and flood the system and get rid of everything. Surgery is limited in the scope that we can do with this disease. But what you're looking at is a little survival curve here, modern data. This is from the Zoom-One setup that we have, a big data set that's pretty good setup, about 418 patients. So to check on 5-year survival, 62%. There we go. Oh, just like that. See, about 62. Sorry for my little chicken scratch there. But about at that a number of months, still about 60% of people are doing well. And do you notice anything else about the curve? About 50% of people, long-term remission, otherwise known as the fancy word cure. Yeah. So anytime you get somebody who's like, oh, there's a cancer cure out there, trust me, we can cure a lot of cancers. There's just a lot of types of cancers, and they have different pros and cons. So what you're looking at is the success of probably 100 years of lymphoma medicine. A setup right here of a chemo set. That's the CHOP right here, these combos right here. And the R is that rituximab. And look at that. You all have heard of drugs that are actually antibodies, right? Same things that are getting these. It's kind of neat. Seeing antibodies kill B cells that we targeted them to find, CD20, an oncogene, and kill. Rituximab's awesome. We have long sought to deal or to get anything better than this. And as long as I've been in the field, nothing's changed a ton. It's a really reliable regimen. It's good. Now, should you be one of the ones that does fall in this curve, one of the 40% in 5 years that does relapse or refract the therapy, now, oh shoot, I dropped my line too quick. Dang it, sorry. Got to go back to the DNA sequencing. Given this and why we know this and why we knew to target CD20 is once we had the genome, we could start seeing what the proteins look like. Once we knew what the proteins look like, we could design organic molecules that killed those proteins, competitive or allosteric inhibitors. Bonus points to the farm people out there. So in this case, you're looking at one of the main driving force pathways, the BCR pathway of B cells. Remember that this is a natural good thing. Remember that that thing wants to bind the right pathogen, right? Grow and survive. And if kappa B right there, the natural process that just gets corrupted. And when this gets corrupted real bad, and you end up as one of these patients that doesn't do well, that's what happens next. Now at 2 years, only 20% of people are still around without an event. And an event can be side effects, death, pulls out of the trial, any sort of bad thing that happens. So this has been the other side of the field. We have never been able to overcome this setup right here. This paper from 2017 was essentially the kind of one of the big calls to action that we made no progress in the 10 years before that. This is what we call RR DLBCL, that second stage, that if you do get rid of R-CHOP, you're in this population. Something else happened. Instead of just regular drugs, we made villains, we made monsters of our own, to say the least. There was an idea, you guys are all smarties, so you can kind of walk through this. I'll tell you a story where it came from. At Penn Medicine in Pennsylvania, there was a doctor, Carl June. He had a leukemia patient. She was about 6. She was completely toast in terms of the genes, where tumor was at, it was over. He had heard of a therapy practiced in the Middle East and in Israel that had been researched where you would take out a patient's T cells, immune cells that kill things. They're the same cells that kill tumors inside you when they grow. You would take them out and you would genetically modify them. You would GMO the patient's T cells. You would give them a synthetic gene that didn't exist in nature that would kill an oncogene, CD19. That was the idea. This has only been ever happened in cells and mice. You would expand these, you would transplant these back into the patient. So June does this. And at first, the girl actually kind of recovers a bit, but then a catastrophic neurological delirium happens. You guys know the big connections between the immune system and your brain, right? The reaction of these CAR T cells, chimeric antigen T cell receptors, the synthetics, was so great, she basically lost her ability to think for about 2-3 days. And June actually signed his resignation because he had thought he had basically killed the child. And that, it was kind of a, you guys remember the Challenger rocket ship explosion? You heard of that? He was like, there was going to be that moment, and this would be over forever. It's therapy. But child came out of the delirium. Child came out of the cancer. And to this day, she's now 20. She cured it. The new curve for RR DLBCL is at 24 months. That's closer to about 50%. We went from 20% to 50. We get a Nobel Prize for going up 10%. So that was fun. I mean, this was a huge landscaper deal when this happened. And I can go back to the conference I was at. I think it was about 2019, the one before COVID happened. CAR T was in the game, and everybody kind of thought, oh, this is this year's fad. This is this year's, metabolism type stuff. Yeah, who knows? No, now we've got it rolling now. And what the other thing that we've got is we've got so many patients and such cheap sequencing and so much defined what's a bad gene and what's not in lymphoma, that we've been able to basically cluster these patients into 5 major genetic groups, essentially. Those of you that think in phylogenies, this is kind of your time for that. These patients, and not only that, genes themselves naturally cluster into about 5 groups. The groups, the scientific groups, they all kind of argue. That's why there's 3 different names for each one in some cases. They're all still in a bit of a big fight right now. But the good news for all the rest of us is that each of these groups has some pros and cons, strengths and weaknesses, and each one is vulnerable to specific drugs. Each one has a different susceptibility to the main living drug that we use now, the CAR T cells. Some of them respond very well, but others not so much. Here's where we kind of come in, and I know I've been talking a little bit. I'll speed up a bit. We want to know, based on the genes, who is not going to do well with CAR T. Why would we want to know that? You don't have to shout anything out. How would we want to find patients that wouldn't do this, or that would be already predicted to fail? How expensive do you think it is to have a team of scientists for 14 days genetically engineer immune cells? $100,000? $250,000? $500,000? $500,000? That's about as cheap as you can get it. So if there are organic molecules instead that can do the job for us, we avoid not only the toxicity with the brain stuff, which we are controlling still better, but something new in the field, financial toxicity. Therapies that are so expensive that it's almost, there's a big debate right now in the field if we can even keep going this direction if nobody can afford anything. So hence why we want to find this population. So in the beginning, there was my napkin. Who does bad with CAR T cells? Here's where all roads eventually led, is that if you had a patient that what we would call an immune cold or a depleted tumor microenvironment, that's TME. If you were a patient that had no T cells around and it was like a desert, if you sent in CAR T cells, it'd do nothing. It'd be like sending in soldiers without ammo, basically. We also found that there were connections between certain genes and certain pathways that would portend and predict that cold desert environment CAR Ts would simply die off in. Basically, who was already set up to fail with these, that became our goal. That became the niche that we could try and find and predict before the other bigger labs could do it. This is just a better version of that. I didn't want to make you look through that thing. On the napkin, I've even lost the color to what the drugs were. But most of these roads lead somehow over here. Some of these roads inform each other. Some are a little more unique. But in this case, we wanted to take a look at a few major sets of genes. We started pretty big. You guys will actually probably know some of these gene names. Here's the first one, p53. Of all the genes in your genome, this is the one that protects your cells from going bad the most. If you lose 1 copy, it's not good, but if you lose 2, the races are off, like the guardrails are off. That cell can do just about whatever it wants. So we're starting pretty easy going here, but again, this is a new patient population. So of course we kind of isolated. Let's look at one of the major genes for this. So here's some fun data sets. My sources, quote unquote. We are very excited to have all these. But let's take a look. If we go into one of the initial data sets, Zoo Monet 2022, on the left, a survival curve. If you just have p53 in general, here's just a good background look. That's not good. You do not want to be in that red curve. And then, but equally on the right, this is the regular R-CHOP treatment, but this is with CAR T. You go from about a 2/3 chance of doing fine if you don't have p53 to a reversal if you do. So that is not an ideal place to be right there. Writing was on kind of on the wall for this. So starting from a bit of a hypothesis generating standpoint, what else is associated with p53 loss? So I'm going to shorten it to p53 in this case, so I don't have to keep saying TP. But we also wanted to say, okay, what else is associated with survival? And in our case, EFS24 is what we counted at Mayo was if you failed before 24 months, you entered a very bad population as a patient. So pairing that, we actually ended up finding that when patients were altered for P53 and when they failed, there was one gene that shined all above the rest, BRD4, or as my wife Emma calls it, bird4, which I find is insulting because it doesn't sound as scary. That is an oncogene right there. And as you can tell right here, when you have bird4, you're in that red curve again. Still significantly not ideal. What this setup in fun colors is going to show you here as well is what pathways are increased in these P53 tumors and which are decreased. So you can kind of tell some of the green ones that are helpful for growing, some of the metabolism ones that are helpful for growing are higher. Some pathways obviously also lost some stuff. Data's data, you're not going to throw them out just because they're unique. But one set were lost in both cases. Remember how I warned you, cells that can't die? These are the genes that help cell death. They're gone. These are the genes that are recognized by the immune system. They're gone. So you are sitting and flying blind if you are one of these patients, statistically speaking. Here came one of the best papers that we've ever done, I think, in the field. So Ollie, oh shit, this is awesome. His name is actually Ollie because he's from Norway. Ollie Durva did CRISPR screen. He knocked out every gene individually and then said how they do against my synthetic CAR T cells. Here's the top genes that he found were required. These were needed for CAR T success. These genes right here are highlighted right here. TNFRSF110B, is a death receptor. Sounds pretty cool. T cells need this to bind to, and then they kill the cell. So if you send in a CAR T cell, and this gene is gone, T cell just runs around not killing. Shoot. That is, oh no, I hate when this happens. Sorry, I hit the home button on that. Ignore that person. Sorry, this is what I get. Yeah, Greg, you were right. I shouldn't have relied on the technology so much. We'll get it back. That happens like once or twice in genetics every year. All right, minus. There we go, and come on. All right, back in business. I'm on back. Sorry. All right, good. Anyway, back to our fun death receptor story. Here we go. Yeah. So we talked about that bad oncogene, BRD4, right? There it is. That gain and that loss tied up as the X and Y axis right here. Lo and behold, what genes showed up as the double loss in both cases? Death receptor lost in both cases. Left and right, X and X. CDKNA1A. That is a key tumor suppressor. Remember mitosis and all the cycles, G1, G2, S phase, all that? That thing stops those. So that's lost too. Not ideal. So we're looking at a pair of losses here that are pretty devastating. Durva's score right here is saying how his score was like, this is a massive need for the CAR Ts to work. And it was lost in each case. And CRISPR-KO score just confirms these are essentially tumor suppressors in that case. So here's where things get real fun though. We can take the patient data, even though we don't technically know exactly what immune cells were hunting and running around their tumor, but we can take the raw data, feed it to a machine that does deconvolution. And what that does is it infers from the genes that were there what cells were running around that environment. Were there T cells, were there neutrophils, were there macrophages? Before we even get to that, even if we just look at some immune strength pathways like interferon, STAT, we see that those with P53 way down in that red, right? Not ideal. So low immune systems for them. But what's more is look at the low immune power of anybody who lost that death receptor, that TNF gene. They're also in the same bad boat. And each time we looked at these with P53 as well, these are 8 fun immune genes in this case. Each time didn't work out super well. So when we take a look here at the deconvolution, we're going to go from the land of genes to the land of cells here. We've got about 22 immune cells that we can deconvolute. For a sanity check, up there is B cell memory B cells. So they're highest in the low TNF and the low P53 group, because that's the cancer cells. That's the cancer we're studying. So there's our sanity check number one. But then in the lower left, again, who's our double losses? CD8 T cells, those hunt tumors for you. Gamma delta T cells, those are going to hunt tumors for you, and they're gone. Macrophages, before they develop into bad ones, M0s, those could have been helpful, they're gone. Not going to lie, neutrophils showing up here, a little weird. We're going to figure that out someday, but the data's the data. There they are. Among some of the other guys, there are, for example, bad guys like macrophages M2 that were higher in those low death receptor ones, but lower in the P53 strangely. So going just to straight up survival though, because the clinicians love survival. You gotta have survival curve if you're going in any medicine or science. Anybody with a P53 alteration, we also check the status of their CD8 T cells. Were they there or not? If you did not have a presence of CD8 T cells, you're double toast. Remember this curve from the P53 people, how bad it was? Almost twice as bad if you are also in an immune desert of depleted TME like we saw. The same pattern held right here for that TNF death receptor. Should that, in those low patients, those that were basically about one standard deviation below the average, classified them, they lacked a T cell presence of any kind. There's a split. And that's the key with these black curves right here. These are still ones that are altered for the thing, or for P53 or TNF in any case. They're just double bad because they also had that depleted immune system. So going back all the way to the goal, which was find the patients that would not do well with CAR T. We're finding them, trust me, right here. The networks that BRD did as well. I haven't been as fully honest about BRD yet. It is an oncogene. Remember from genetics, what is the most pleiotropic gene type there is? Transcription factors, bromodomain inhibitor, right? Anything that is an epigenetic master regulator. BRD is one of those. It is responsible for turning on thousands of genes. So when it is out of control, we see a massive setup of programs that are going to show for growth, survival, the whole shindig. Not only that, it negatively correlated real bad with that good tumor suppressor we had, CDKN1A. We saw that if we just isolated, for example, CDKN1 interactants, when people lost that, the main perpetrator, when they would lose that tumor gene, was our friend BRD4. Again, kind of a big perfect storm to say the least. And one last connection, the TNF gene, the gene that it most correlated with of all genes was the same tumor suppressor, CDKN1A. So there is this sort of network between the death receptor connected to the tumor suppressor, also connected to P53, and also a relationship with that devastating oncogene, BRD4. So we had sort of begun to define our patient population. And of those four members right there, one of them is an oncogene. And we can drug oncogenes, right? We can kill it. The other four are gone. And that's where something else that was really fun in this project happened. We had one oncogene on the table to drug and kill. That's typically not enough. People like combinations. And most drugs cannot bring back a mutated gene. Except one that we had heard about in about 2021. P53, I promised you, right? Big deal gene. When it's mutated, it's bad. It folds up in a very awkward way. But an experimental drug came out through pancreatic cancer research, worked its way into my ASH meeting, my hematology meeting, in one of the macrophage cancers. APR246, otherwise known as aprenopopt, refolds or prevents the bad version of P53 from accumulating. It pushes it back into the right protein folding position. At least that's what it's designed to do. I was skeptical, to be honest, because I'm a big drug kills gene kind of person. I wouldn't even call this an agonist, technically. It's like a fixer, basically. So strange. We gave it a shot, though. It worked. This was the first time we'd ever tried this in lymphomas. So we did that, and we did a more established gene called Edas-Nutlin right here. See how this little MDM2 gene kills P53 naturally? Why not drug the negative regulator of our good guy, right? So a pretty easy setup with that one. And sitting here, here's our aprenopopt, and we're boosting SP53 boosts our death receptor. And we said, look at that. Looks pretty good. So in black is aprenopopt, white is the MDM2 inhibitor. And they both do pretty well against our cell lines. And again, this is always really fun because you get to see the cell lines all like collapse and die and everything. But in any case, it was fun data to say the least. The most fun was the Western blot. So now looking at this image the second time, who's that gene right there? The actin is just a control. It's just supposed to be there at the same level. Guess who came back from the dead once we drugged it? We brought the death receptor back. Bring back P53 and limit its destruction. We're back in business. Back in business. So that was pretty fun. Setting that up. That was kind of a position on this one was, this was the first time that we had said, maybe you don't have to drug these patients to kill the tumor. Maybe you could set them up for success against the CAR Ts though, right? If you can bring that death receptor back that the CAR Ts eat and kill, it's a good start. All right, next, we wanted to kill these things. All right, so let's say the CAR Ts are not gonna work. Based on our patient profile, we wanted to set up a fun synergy drug trial and say, hey, let's actually destroy these things. And we found a nice one right there. PROTAC, ARV-825 was recently developed. And a PROTAC's unique because it actually destroys the protein itself. It doesn't just like plug an active site, demolishes it instead. And then we had an excellent drug called abemaciclib. This is actually on a lot of drug commercials right now. I think it's bereslilib or some fancy name that they're doing now. We have not ever, CDKs are drivers of mitosis. So every time you saw the S and G1 phase, that's one of those common ones. It's been a very hard thing to create drugs that can actually get those. And we were there. So there's those standard curves up there. But here is where we get synergy. And what that means is that if you killed 20% or if you killed 10% with drug A and 5% with drug B, and you put them together, and you killed 15%, that's just fine. But what we were seeing was that they were killing 80% instead. 60%. And then when you put them in combination, you sort of corner the evolution of the pathway. The tumor cells had nowhere to go. The red indicates areas of high synergy and areas where the drugs were essentially killing more than you would have predicted statistically that they could. And then we went into the microscope. DMSO is just a control. It's what we put the drugs in. There's abemaciclib alone, still a big old mass. ARV alone, and then right off the map. So we had a pretty good time with that. Now, unfortunately, sometimes science is a little ahead of the old clinic there, and we get too big for our heads. What do you think the problem with PROTACs is? They're pretty cool. See them right here? They actually link whatever their target is to your natural destruction system in your cells to get rid of stuff. That's awesome. You actually destroy the oncogene. But see this? It's going to go back and forth over and over again. It's not going to get degraded. Most drugs cancel out one for one, so you can dose pretty easily. To say the least, these are still in a bit of a phase 1 trial mode. They're pretty volatile in humans. The other caveat, BIRD4 helps your T cells, your good guys. Isn't that terrifying? That a lot of some of the bad genes in some cells are the ones that are trying to help the good ones. So the good news is getting those things out here was a really cool step to say, hey, the synergy works, number 1. Number 2, PROTACs can work as single agents. Number 3, will be worth to keep looking in on this. Still. All right. Same little guy again here. We had another gene that we wanted to investigate, this time an oncogene. This one's a little simpler of a story, but still fun. And hey, the figure slapped the first time, so I didn't change the colors up too much. I'm sorry, call me lazy. But let's target another gene in lymphoma. Story number 2, MYC. This is a transcription factor that is like grow, survive, let's go. Pretty common oncogene, but again, hadn't been investigated for the population we wanted. There's your trajectory in normal lymphoma, and there's your trajectory with CAR Ts. And we'd actually added more data to our big CAR T bank at this point, so significantly not a good time to have this be high or be mutated. Not a good time. But like I said, the game is changing in this case a bit because MYC is an oncogene. Here's the things that should be high with that oncogene. We see a bunch of cell growth stuff. We see some kind of mixed bag on cell death and things like that. But the one that wasn't mixed again, if you have high MYC, you are existing in that immune desert again. Your signals are down, you can't be recognized or killed very easily. There is an opportunity here. Problem is MYC is in the nucleus. Getting drugs into the nucleus is actually really tough. Drugging MYC itself is actually really almost impossible. It doesn't have a good spot to it, essentially. There's one thing that we could. To get MYC out of the nucleus as an RNA before it can actually be born as a protein, it requires this little guy, exportin 1. We can drug that. See that in a bit. But back to some devastating gene losses. If you have high MYC, standard deviation or above, we actually paired this with new data from Natalie Schaller's group, who said, we have gene data that says who's high and who's low when CAR Ts fail. So this was new data that we hadn't had in the last analysis. We paired it this time with our setup. So look again, right here. Who is low in both cases? Who's low when you have this bad oncogene? Left. And who's low in the people that don't respond to CAR T? This is the first time that we had actual CAR T RNA instead at that next level. And here's what I'll tell you. T cell gene, T cell gene, T cell gene, immune gene, macrophage gene, NK cell gene, T cell gene, immune gene, immune gene, immune gene, immune gene. Sorry, I'm going to keep doing this all day. I can do this all day, Captain America. All right. So the losses were horrible. It was a bloodbath for the immune system. Again, if it's not there, it can't connect to the CAR T cells, you're toast. Similar profile again. Set this up at this time again. We wanted to go for the cells themselves. Lo and behold, again, here's our CAR T failure rate and association with each cell type. Our association type, if you had high MYC. And again, unfortunately, there's our friend, the best friend against the tumor, your CD8 killer cells. They're done. They're low and low. So again, finding another population that even if you sent in a bunch of CAR Ts, if there are not partnerships waiting to be revived by them, will not work. And the cool thing with CAR Ts, they also become the commander CD4 types. They need an immune system to command. And if it's not there, you're toast. This one was interesting. The reviewer actually pointed out something pretty interesting on this one. The main focus on this was for me to say, hey, if you have high MYC and you don't have any CD8 cells, bad. There's the red curve right there. It's significant against all the other curves. This goes back to the weird dichotomy that these genes play. In that teal curve, the reviewer pointed out, some tumors had high expression and high levels of MYC in their environment, but also had high CD8 cells. Maybe not significantly, but he or she did point out they did better than everybody else. So the T cells that actually have higher MYC potentially killed better. See how hard this is? There's no winning here. Ugh. So here's, this is not on the exam, don't worry. But where the research is sort of heading that's neat is that it is less about how I was trained in my PhD about what are the genes inside the cell versus what is the environment around it. The environment drives the evolution more right now, we think, maybe, than the organism itself, the cell. That's kind of hard for me to accept still, so I'm being stubborn, but so far, so good on a lot of new studies that are really helping us see these new ecosystems, ecotypes as we're calling them. But here we go. We had some of this data, and we can see here are, if you are CD8 absent and you are high MYC, so that was that really bad setup, that red curve, you have less cell activation, less T cell activation, less lymphocyte differentiation. Again, losing as many possible immune setups as you can significantly. Not gonna lie, these are just fun XY plots that I made pretty. I can't throw them in the trash though, they're just fun. But I did want to see, for example, the MYC oncogene versus the CD8 gene itself, X versus Y. And you can see that negative correlation right there. The cell states are those ecotypes. And if you're a larger bubble, it means you failed. So it is a fun little expose. And I don't know, I'm part of Graph Head Prism Mafia, so I had to show these off. I'm contractually obligated to. There's another immune gene, negatively correlated IL-7 and CD58. a natural killer cell gene. Some of the minions right now are working on that one. So, speaking of head minions, the reviewer asked for something called single-cell RNA-seq. Instead of just a big bulk sample of a tumor, and then pull it out and get RNA out of that, you would feed each cell through, and each cell has its own set of RNA up and down. So Adam, in his computer genius, was able to harness these data because I saw them and nearly came to tears. But given this, he was able to find patients with higher MYC, showcased all of these setups. A higher, is correlation the best word for this? For now, yeah, let's do that. More correlation with these high mitotic cellular stress type of stuff. Look at some of these, IL-1, IL-8 production, transforming factor, TNF, all these guys. We found all kinds of cool data. The reviewer was like, I don't know if we can accept this project. There's not any single cell RNA-seq. And I was like, oh, just you wait. Figure 4 is here to arrive. So, thanks, Adam. Good job. Moving on. Okay. But we still wanted to drug some stuff. That wasn't enough. Genes and data are too boring. Let's kill these things. Remember MYC? Let's take this guy down. MYC really helps. A CDK9 right here. It's one of your drivers for mitosis. My friends at AstraZeneca recommended their new drug, AZD4573. It's going to kill that target. So we're going to hit it from the front. And remember this little guy, XPO1. This helps MYC become a true gene. We can't kill MYC itself. We don't have the drugs to do it. Doesn't bind. But we can kill its partner. Selinexor, another drug that will help us here. So if we go here and we go here, we can defang MYC, hopefully, was the idea. And lo and behold, like the worst magic trick ever pulled, of course it worked. And notice something about these drugs too. Like when I started, I would typically see drugs falling off a little bit later. Notice with AZD in the red, even at 78 nanomolars, it was killing it. I used to have to see 100 times that to see that when I was my PhD. So drugs are getting so much better at killing these things. Now, as you can tell though, it was kind of a weird little response. These don't look like the curves we just saw. They're a little immediate. They're a little choppier and angrier to say the least. But what we do? Slapped them together, saw what happened. These were higher synergy rates than the last one. And I still do need to send, well, I did present these data already, so I don't know, I think maybe AstraZeneca is going to scoop me on that. But we're in review right now, so maybe we can beat them. We can be like the small sneaky fish that like gets in and steals from the bigger one. Now, here we are. We've got some cool projects going and kind of accomplished in a lot of cases. We found P53 and its connections. We looked into high MYC presence. We're currently looking in how metabolism, everybody's favorite thing, like glycolysis and ETC, is playing this role in a depleted environment. Things are getting more complicated, though, for what this response actually is. And ask Adam or Diego, there's all kinds of new things coming out. One of which is that it is bad to have 0 immune cells around. But unfortunately, things can also get a little too hot sometimes. And that's bad for CAR T too. So the balance is strange. So right now, as it stands, there's a new villain in town as well. M2 macrophages shut down your immune response. They literally eat the other cells and kill them. Kind of violent. Where we would like to go is join this little crowd right here. Instead of drugging the patient tumors anymore, what if we drugged the CAR T cells and made them go crazy and kill higher and better? The last one is just kind of a fun story that single-cell RNA realized. People that actually have responding tumors, they have about 5 to 10% of their B cells are not tumors. They're still trying to do the right thing and they fight back, like a little guerrilla force, essentially. Kind of neat, but I don't know what actions we can take on that quite yet. It's pretty deep sequencing stuff. But we tried something. We did this in genetics. You guys recognize this? Yeah, I Watson and Crick some of your results. So what we did, I'll give you that. Oh, that was a bit of a dig, yeah. So we had some macrophages in lab, some nice healthy, there you go. We treated them with an epigenetic modifier. This gets rid of a methyltransferase, turn on and off a bunch of genes so we can maybe the macrophages will go crazy and kill the tumor. And there's our tumor cell right there, DHL4. This is our preliminary setup. There's the cancer cells alone in their replicates. There's the tiny amount of macrophages, so it makes sense that they weren't really alive a ton. There's the macrophages paired with the tumor, but no drugging, no craziness. But if you added the drug, and we pull the drug off the macrophages after an hour, but we change their gene profile, there it was. So we are acting on this. And this is only the beginning, Paul Atreides. We have so many more tests to run and other stuff. It's going to be exciting. Don't worry. And well, I'm not going to spoil Doom, but anyway. So we've had a fun time. We do our best here at Little Mankato, but I think we've had a good setup here. I'm kind of revealing my color scheme a little bit on the posters up there, a tad. But we do have another one in review right now. That's the MIC project right here. Here's the P53 project. Here's one of the old projects I did when I first got here. So we stay busy. It's been fun, to say the least. And then, yeah, some of you know the other fun studies that I think, yeah, that I do. I track the hours working and run around and see how you're doing on your exams and such. Don't judge me for my average exams. Just look at the pretty colors and enjoy. Oh, right. Oh, come to class. Look at that. Very naughty students. All right. Oh, yeah, we don't have to do that much. Don't worry. I'll save the bioed stuff for later. All right. Anyway, thanks so much for listening to me share our fun projects.